Well, we've been looking at the transcription of RNA polymerase II encoded genes. In contrast to RNA polymerase I and RNA polymerase III, the RNA polymerase II encoded genes encode messenger RNAs. And of course, the goal of the messenger RNA is to ultimately make a protein. And then from that protein, we get a new function in the cell. So the gene is turned on in order to generate a new function in the cell. So we've seen all that activity taking place in the nucleus, which is the transcription, the capping, the polyadenylation, and the splicing. And now we're out in the cytoplasm where messenger RNA takes place at the ribosome, so that the information moves from the DNA and gets transferred to the ribosome in order to synthesize the protein. So we're going to look at messenger RNA translation. And there's three aspects that I want to focus on with respect to this. And the first is I want to look at what the messenger RNA looks like. We know it has a 5-carbon cap. We know it has a polyadenylated tail, and so on. But there's more there. Just like everything else, there's nucleotide sequences that are part of the RNA that control it. Just like there's part of with the DNA, and it controls transcription. I want to look at the messenger RNA. I want to look a little bit at transfer RNAs, and then we're going to look at the ribosome just a little bit, and then we're going to put it all together and see the steps that take place. So again, we're looking at translation of messenger RNA into protein. We're going to look at the messenger RNA, tRNA, ribosome, and then how it all fits together. And the first thing I want to look at is the messenger RNA. And of course, the messenger RNA dictates the amino acid sequence because the messenger RNA contains the proper codons that bring in each amino acid. It does a few other things. It has to set the reading frame. There's 3 different reading frames on the RNA on any nucleic acid. And we'll look at that in a second, try and explain what that means. It also signals where to initiate translation. It's not at the very beginning of the messenger RNA. It's not at the end. There's certain sequences that dictate where things should begin. So first of all, the reading frame. If you take any nucleotide sequence and you choose a set of 3, you can call that a codon. And then the next set of 3 would be the next codon. And the next set of 3 would be the next codon. Well, if you look at what I've drawn here, I have a sequence, GCC, AUG, AGG. So I am setting reading frame number 1, there's 3 there, reading frame number 1 with the AUG as the first codon. I'm doing that for a reason, because AUG actually always is the first codon. So that's one of the ways that the sequence dictates the reading frame. So the first AUG is codon number 1, and then the next codon would be AGG, the third codon would be UAC, and so on. But any 3 nucleotides can be seen as a codon. But in this case, only one is the correct initiator one, and the second and third. So let's look at reading frame number 2, which is on the top. What if it started at UGA? So then the next codon would be GGU, and the next codon would be ACU. So that's different information. There's a third reading frame. That is, if you start on the bottom, if you start the first nucleotide of the first codon is G, GAG would be one codon. The next would be GUA, would be the second codon. The third would be CUU. So there's 3 sets of codons, in a sense. 3 what we call reading frames. Each one, in principle, could encode a different protein. That's usually not the way it works. Usually one reading frame is sufficient to encode the protein. So that's the first thing. So the first AUG generally sets the reading frame. Now if we look down at the bottom, we see a line drawing of a messenger RNA, the 5' cap, polyadenylated tail at the end, 3' end. And let's look at what we have here. So the first AUG is the initiator codon. Occasionally it could be the second AUG, but it's most 99% of the time the first AUG encountered sets the initiator codon and sets the reading frame. So AUG encodes methionine. Now there can be more methionines in the protein. AUG is the initiator codon, but AUG also encodes methionine. So you can have additional AUGs in the sequence, and so what I'm showing here is there's 3 AUG codons, so there would be 3 methionines. The first one would be the initiator AUG. So it initiates at that AUG, and there's that line there. You have initiator AUG, and then you have the line going across with an arrow at the end, and there's the stop codon. And that ends translation. Now, if you notice, the first AUG is not at the very beginning of the message. And in fact, the stop codon is not at the very end of the message. So it's more towards the center of the message. And so the sequences that border the start codon and then the sequences that border the stop codon are called UTRs, untranslated regions. So there's a 5' untranslated region, 5' UTR, and there's a 3' UTR, 3' untranslated region. These do not encode any protein. They are involved in regulating the availability of the transcript for protein synthesis. So they usually are somehow involved in regulation, just like a promoter is involved in regulating transcription. These elements, each RNA has its own regulatory sequence. Sometimes the 5' UTR is called a leader sequence. And that 5' UTR can be anywhere from, let's say, 100 nucleotides to even as much as 500 nucleotides. And the 3' UTR can be, again, anywhere from 100 or so to 500. So they can be very large, and they play a very big role in how this messenger RNA behaves. Now, in addition, there's another sequence that I've written there, which is just upstream of the coding of the initiator codon, and that's called a Kozak sequence. This was named after the woman, Marilyn Kozak, who identified this by sequencing hundreds of messenger RNAs, and she came up with this highly conserved sequence just upstream of the start site. And that sequence is GCC, 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 basically a run of GCCs. Be a repeat of 2, 4, 6. And what that does is it helps the ribosome identify the initiator codon. So it's a signal, part of the 5' UTR. The way it does that, the way it recognizes that, the ribosome recognizes that, is through base pairing with the ribosomal RNA that's in the small subunit of the ribosome. So again, base pairing interaction. So that's the overall structure of the messenger RNA. Let's move on and look at the transfer RNA. So transfer RNAs are tiny RNA molecules, about 70, 75 nucleotides. They undergo a lot of internal base pairing, and you can see here, and so they form this kind of what they call a cloverleaf structure. It's not flat like that, it's three-dimensional. So you can see the base pairing, GC, CG, GU, AU, and so on. Often there are unusual nucleotides in these transfer RNAs, which are not found in other RNAs or other DNA molecules. And you can see some Greek symbols representing some of these unusual nucleotides. They just have slightly different bases attached to it. So they don't undergo base pairing. Well, it looks like maybe they do, because it looks like the A, right towards the bottom, looks like that A base pairs with that other one, that Greek symbol. I'll look that up and I'll get back to you on that. Okay, but the bottom line is undergoes base pairing, internal base pairing. The very bottom part has a 3-nucleotide sequence, which was referred to as the anticodon. The codon encodes dictates the amino acid, and the anticodon base pairs with the codon to bring in the proper amino acid at that location. I'll come back to that in a second. And then at the 3' end, remember there's a 5' and a 3' end just like any other nucleic acid. And the 3' end always ends with CCA, and then the amino acid is attached to that adenine at the 3' end. The amino acid is attached through a bond with either the 2' or the 3' hydroxyl. And there's an enzyme that carries that out, which is called amino acyl tRNA synthase, which we'll look at in a second. So that's the transfer RNA with the codon, has the amino acid, so it's kind of like a hybrid amino acid, nucleic acid. Now, what I want to point out is, look at the codon. So if you were to look without thinking about this too much and looked at the one on the left-hand side, you see the codon, anticodon, GAA, you might think that, oh, okay, that's pretty easy. The codon is CUU because CUU base pairs with GAA, but that's not true. It's not true because I have written down there CUU is not the codon. Why is that? Because we always draw the nucleic acids in the 5' to 3' direction. So if you look at the transfer RNA, 5' is on the left and the 3' is on the right. If we write out the codon, we also write it out 5' to 3', but that's not the way nucleic acids base pair. They base pair in anti-parallel manner. So it has to be flipped. So that's what I have drawn on the right-hand side. I actually flipped it around. So the 3' end is on the left-hand side, the 5' is on the right-hand side. This is probably the way it actually works. Just because we draw it 5' to 3' doesn't mean it has to position itself 5' to 3'. It has to position itself in an anti-parallel manner. So on the bottom there I have a 5' to 3', that might represent the messenger RNA. And so you'd have UUC as a codon, and it would base pair with the AAG because now they're in the proper orientation. Now this transfer RNA with a single amino acid attached to it at the 3' end is referred to as an amino acyl tRNA. It's a very important term. Amino acyl tRNA represents one amino acid on the transfer RNA, which is generally how they exist. Now, there's a very important step in this, and that is, the correct amino acid needs to be added to the correct transfer RNA. If there's a mistake in there, then you get the wrong amino acids put in, and that would pretty much be lethal. So there's an enzyme that kind of is really, in a sense, the ultimate translator of the genetic code. That is, it reads the GAA or something else about the structure of the transfer RNA, most likely the GAA, and then knows which amino acid to attach to it. So it's an absolutely crucial step, and that's carried out by an amino acyl tRNA synthase. So the amino acyl tRNA synthase, they're enzymes. There's 20 different of these, one for each amino acid. So the amino acyl tRNA synthase couples the correct amino acid to its corresponding transfer RNA, and that process is called charging, so charging the amino acid. So how does that work? Just in a very, simple way of showing this, the aminoacyl tRNA is an enzyme. Here we're looking at one that does tryptophan. So the transfer RNA, so there's two binding sites on this enzyme, one for the transfer RNA and one for the amino acid, and then there's an enzymatic activity that then links them. So here it shows the binding site. There's a binding site for the tRNA, there's a binding site for tryptophan, and then there's a reaction that takes place, which is the synthase reaction. Uses ATP, so there's an energy step there. And what you see next in the middle is the amino acid is now bound to the transfer RNA. And it's bound, I have it written on the right-hand side, it's bound to the 3' hydroxyl of the transfer RNA, sometimes the 2' hydroxyl of the transfer RNA, but it's always the very last nucleotide. And then, of course, that charged amino acid, which is now carrying the amino acid, can now base pair with its codon. Now, this is actually a remarkable enzyme because it has to choose the correct amino acid and the correct transfer RNA. And the thought is that it completely engulfs the transfer RNA, and it's recognizing, maybe even contacting the anticodon, which then permits it to then bring in the correct amino acid. Now, before, when we looked at the chart of the amino acids, the genetic code, there's this what's called degeneracy of the genetic code. So some amino acids have 4 or sometimes 6 different codons. Yet, this aminoacyl tRNA synthase, there's one for each amino acid, so it will recognize all of the respective transfer RNAs. So that means it would recognize 4 different transfer RNAs, put the correct amino acid on. So this is really ultimately translating the genetic code. And so they have to be very accurate, of course, and they're very highly conserved, because any changes, any mutation in any of these genes that encode any of these synthases would simply be lethal. Any mutation in an aminoacyl tRNA synthase would be lethal. Why would it be lethal? Because it would put the wrong amino acid on in every position in every protein. Obviously, every protein would be messed up. Okay, so that's the transfer RNA. Now let us move on and just look a little bit at the ribosome. The ribosome is an enormous complex. Actually, it's a ribonucleoprotein complex. Why is it a ribonucleoprotein complex? Because there's RNA molecules and protein together. So the eukaryotic ribosome is a large complex of 4 ribosomal RNAs and more than 80 different proteins. And they come together to form these large subunits. There's what's called a small subunit, and then there's a large subunit. The small subunit has 33 different proteins, 1 ribosomal RNA, and that's called 18S ribosomal RNA. That's 1,900 nucleotides. The large ribosomal subunit has 49 proteins and 3 ribosomal RNAs. There's 2 small ones and 1 big one. There's called a 28S, which is large, 4,700 nucleotides. By the way, that 28S ribosomal RNA is the ribosomal RNA that carries or expresses the peptidyl transferase reaction by virtue of the RNA reactivity. Links the amino acids together. There's 5.8S, which is 160, and then a 5S, which is 120. So there's 3 there. These 2 subunits come together when they're synthesizing proteins, and so that's what's shown below. complete eukaryotic ribosome, large subunit, small subunit, 4.4 million 200,000 molecular weight. It's just, really big, lots of proteins, lots of RNA molecules. Now there's binding sites for RNA, transfer RNAs, ribosomal RNA, transfer RNAs, messenger RNAs in the small subunit of the ribosome. So let's look at that next. So on the left is a ribbon diagram of a ribosome, and I think the colored, the red, the little orange, and the yellow may represent transfer RNAs that are in there, but I'm not sure about that. But anyhow, that's kind of this overall ribbon diagram, three-dimensional structure of ribosome. And then we look over on the right-hand side, it has more of a diagram showing it a little bit differently. So we have the large ribosomal subunit, the small ribosomal subunit, and the binding sites for actually 3 different RNA species are present in the small subunit. They extend a little bit into the large subunit. So of course there's a binding site for messenger RNA, because the messenger RNA has to be there. The truth is that it's not on the surface. What it is there's kind of like a tunnel in this structure, and the messenger RNA enters the ribosome and moves through it and exits on the other side. Then there's these 3 sites called A site, P site, and E site. So let's talk about what those are. Those represent, the A and the P represent names of different species of transfer RNAs. The A site represents the site for the amino acyl tRNA. And we looked at that just a minute ago. Amino acyl tRNA is a single amino acid on the transfer RNA. The P site stands for a peptidyl tRNA site, and I'll show you what those look like in a minute. The peptidyl tRNA site, and the E site actually means the exit site. After protein synthesis takes place, after a new peptidyl transferase reaction takes place, then that E site has an empty transfer RNA and it's released from the ribosome. So E site, P site, and A site, and we'll look at what is bound to those now. So this is illustrating kind of an outline of what happens during protein synthesis, during translation of the messenger RNA. So you see the large subunit, the small subunit. We're looking at midstream, okay? Things have already happened. Why do I say things have already happened? Is because we're looking at, if you look at the P site, there's a transfer RNA that has 3 amino acids attached to it. So that orange transfer RNA is actually bound, base pairing, to codon number 3. Codon number 1 looks like it was purple, codon number 2 was yellow, codon number 3 was orange, codon number 4 is blue. Okay, so there's a process that's already taken place. We haven't seen that yet. But the point of the matter is that when you have a growing peptide chain on a transfer RNA, that is called a peptidyl tRNA. So anything that has more than 1 amino acid attached to it, it could be 2, it could be 3, it could be 50, it could be 200. That is a peptidyl tRNA, and you'll find that in the P site. The E site is empty because a previous amino acid has left. So that's the E site. And then we have the A site, which has what says a newly bound charged tRNA. Basically, that's carrying another amino acid. That's an amino acyl tRNA because it only has one amino acid linked to it, which is normally the way they occur. So that's kind of a static view. kind of midstream, but it helps identify why these things are called A site, P site. Now what we want to do is look at what happens during this process in order to generate these different products, and how proteins are synthesized, and how they're elongated at each step after each codon is read. So the translation of protein synthesis takes place in a cyclical manner, which is repeated over and over again every time adding another amino acid. So let's take a look at this. It's a little bit small, but you can, I guess if you're using the full screen, you can see it better. But at the very top is what we just saw. So you have the E site is empty, the P site contains the peptidyl tRNA, and the A site contains an amino acyl tRNA. Now what happens when these sites are occupied in that way, the P site is occupied, the A site is occupied, that activates the peptidyl transferase reaction. And what happens during the peptidyl transferase reaction is that the peptide, which is number 1, 2, and 3, which is on that transfer RNA number 3, is cleaved off of that transfer RNA and then moved on to amino acid number 4, making a new peptide bond between those. So the chain just grew by 1. The peptidyl transferase reaction is within the ribosome, the large ribosomal RNA of the large subunit. So that moves that peptide over 1, okay? And now, to continue, the ribosome has to move down the RNA to the next codon. And the next codon there is like a darker blue. So it has to move down the equivalent of 1 codon. And that step is called translocation. So what happens during translocation is, you can see it, they show it kind of in a 2-step fashion. Requires GTP, requires energy. It's basically an allosteric conformational movement of the entire ribosome. It moves down the message. So what they're showing is that the large subunit kind of moves over the equivalent of 1, and then the small subunit follows and moves over the equivalent of 1. And in doing that, what happens is the peptidyl tRNA, which was transiently within the A site, but all this is happening really super quickly. It's transiently in the A site, gets moved as the ribosome moves over. It doesn't move the transfer RNAs. The transfer RNAs stay coupled to their codons. So when that gets moved over, then the peptidyl tRNA is now in the P site, which is where it kind of needs to be for this thing to continue. And then the E site, so that's the ejected RNA, you see that coming out. That's the amino acid, the previous amino acid, tRNA number 3. And then the A site is not occupied. So all that's in there now is the peptidyl tRNA and the P site. The next step is the introduction of the next amino acyl tRNA. Now, the way they show it in the book is a little bit simplified. They're just showing it coming in. So I added a little bit on there. What they say is newly bound charged tRNA, which is really newly bound amino acyl tRNA, put into position number 5, base pairing with the codon. What really happens is it's actually bound. The transfer RNA does not come in on its own. It comes in bound as a complex with GTP and an elongation factor called EF1. These 3 things together, the transfer RNA, the GTP, the EF1, is called the elongation ternary complex. The EF1 is a factor that participates in allowing the transfer RNA to enter that site. It doesn't happen by diffusion. If it happened by diffusion, it would never work. So there's receptors that bind to the EF1 and position it so that the transfer RNA can come in. Now, this is not predetermined, meaning it doesn't know, there's no way for it to know in advance which transfer RNA needs to go in. Does it need to be serine? Does it need to be threonine? Does it need to be methionine? Does it need to be glutamine? I mean, there's no way. The information is, the information is solely within the codon. So what that means is that during this process, the elongation factor 1 is bringing in these ternary complexes, it's kind of cycling numerous transfer RNAs, amino acyl transfer RNAs through. It doesn't know in advance what needs to be there. So it's cycling numerous ones through, and when the proper base pairing takes place between the anticodon and the codon, then that's recognized and the transfer RNA kind of gets locked in and the GTP is hydrolyzed. So the hydrolysis of the GTP locks the system in. So now we're back to the very beginning, in a sense, right? If you look at the very top, you have 1, 2, 3, an amino acyl tRNA. Now we have 1, 2, 3, 4 with a new amino acyl tRNA. And so the next thing that's going to happen is you're going to get the peptide. There's going to be a cleavage between number 4 and transfer RNA. It's going to get moved over a new peptide bond to amino acid number 5, go through translocation, eject the tRNA number 4, and then bring in amino acyl tRNA number 5, and it keeps going through these cycles at the rate of approximately 2 amino acids per second. So that's elongation. And of course, it's going to continue like that until it hits a stop codon. And we'll talk about that in a minute or two. Now, this was elongation. What we haven't talked about yet is initiation. That is, how does this process start? Because the mechanisms are a little bit different. The strategy is a little bit different. So let's take a look at that. So initiation of translation, this is the very beginning of the process. And what we see, if we look at the ribosome and the amino acid and transfer RNA that is there, shown at the bottom, that the initiator transfer RNA, which is methionine, is in the P site. So, the question is, how did that amino acyl tRNA get into the P site? Because normally what happens is, what we saw is the amino acyl tRNA goes into the A site. And then there's a reaction that takes place, and it elongates the protein, and then it gets the peptidyl tRNA is in the P site. So, I'm sure you've all really stayed up nights worrying about this, really causing a lot of distress. So to alleviate that distress, I'll go ahead and tell you what we think happens. So the first piece of this is that, first of all, the initiator transfer RNA is a little bit different structure than any of the other amino acyl tRNAs. It has a slightly different structure. In fact, it's referred to as the initiator transfer RNA, and if you look, I have written in the middle special initiator tRNA. They usually write it out as tRNA with the methionine, which is the first amino acid, with an I. And this transfer RNA that carries this amino acid methionine will be very different than the transfer RNA that would carry methionine if it was in the middle of the protein somewhere. So that's the initiator tRNA has a structure slightly different that allows it to enter the P site. That's the first thing. The second thing is that it also comes in with a ternary complex. So it's an initiator ternary complex. It's different than the elongation ternary complex. It's a transfer RNA methionine, the initiator with GTP, along with another protein factor called EIF2, which stands for eukaryotic initiation factor 2. Now, the other part of this, this question is everything we saw before when we looked at elongation was that the appropriate amino acyl tRNA was allowed to come into the A site because it base paired with the codon. So that's what locked it in, that base pairing. So here we have, with the initiation step, you see there's no messenger RNA there. What we see is that the ribosome is primed, and it has the initiator transfer RNA in there, even though there's no AUG codon to base pair with. So it's just not the same as what we've been talking about. So let's look at what happens. So the first thing is that the two subunits, the large and the small subunits, are separate. That's how they exist in the cytoplasm unless they're in the process of synthesizing protein. So the small subunit is floating separately than the large. And the first thing that happens is that the initiator methionine transfer RNA is brought into the P site in the presence of this initiator ternary complex, like I mentioned, which is the transfer RNA with the methionine, GTP, and EIF2, that's the initiation factor 2. They have it from the text as translation initiation factors. Okay, but we're giving a little bit more information there. There's GTP and EIF2. So that is allowed to bind to the small subunit into the P site. That's what brings it in, is that initiation factor. What happens next is that once that binds, then it allows the activation of the receptor on the surface of the small subunit to bind to the cap structure of a messenger RNA. So what's showing is that you have the cap structure binding, and so it brings the messenger RNA to the small subunit, and it enters. There's kind of like a protein tunnel. It enters that tunnel, so it identifies the cap and brings it in. So now we have the initiator transfer RNA, and we have the messenger RNA. Now what happens is next is that the messenger RNA is moved down through the small subunit. So what do they say here? Small ribosomal subunit with bound initiator transfer RNA moves along the messenger RNA searching for the first AUG. We call that scanning. So it's scanning down the messenger RNA to look for the first AUG. When it encounters the first AUG, it binds by virtue of the base pairing with the first codon. Also, the Kozak sequences, which are on the messenger RNA, also participate in identifying that as the first AUG, or the start codon. Then there's a few more steps. So here you see up at the top, you have the initiator methionine transfer RNA base pair with the AUG codon and the RNA molecule, the message. At that point, when that kind of gets locked in, that GTP hydrolyzes, and the translation initiation factor comes off, so now it's left there. And then you have the binding, at that point, the binding of the large ribosomal subunit. So once that happens, you have the messenger RNA in there, and amino acid in the P site, although it's not a peptidyl tRNA, it's the methionine initiator tRNA. Now it's beginning to look very much like what we saw before. Because what happens then is that The next amino acid tRNA comes in, of course, with the elongation factor GTP, binds to the A site, and then you get the peptidyl-transferase reaction that moves the methionine over to the next amino acid. And so now we're really back to where we started with the elongation. What would happen next is you'd get translocation. The two amino acids linked together are now a peptidyl tRNA. They would move into the P site. The first one would be lost through the E site, and then you'd have a new amino acyl tRNA come in, and just then basically elongation continues. So that's the initiation step. Now, initiation is actually highly regulated. The EIF2 factor is a regulated factor. That means it can be turned off, it can be turned on. There's other factors that are involved. I'm going to show you a few of those. There's a lot. In addition to the ribosomal proteins, there's these other factors that are binding and participating in the process. And they're highly regulated. So here's just a small list of them. So there's multiple initiation factors. And many of them can be regulated, basically turned off and turned on. So you have the one we have EIF1, which actually helps stabilize the transfer RNA methionine. Then there's EIF2, which we talked about. It delivers that initiator transfer RNA to the P site. EIF3 keeps the subunits apart. EIF4 is the protein receptor that binds to the cap structure. EIF4B, that should be a B, denatures the messenger RNA. Any hairpins, in order for this thing to be translated, you have to get rid of those secondary structures. And then the EIF5 binds to the 5' UTR. All these things help participate in a smooth translation take place. Now, I want to give you an example. The translation can be regulated. That means translation can be turned off and can be turned on, meaning that not every messenger RNA that is there needs to be translated. It can be translated only when necessary. So I'm going to give you an example where we have regulation of EIF2, which will turn off and turn on translation. So we've talked about initiation, we've talked about elongation, and now I'm talking a little bit about how the initiation process can be regulated. So here's an example of regulation of EIF2. Required for initiation. Brings in the initiator tRNA into the P site. So the activity of EIF2 can be regulated by phosphorylation, basically inhibited by phosphorylation. So I have on the left-hand side that little drawing that represents EIF2, that circle, and that's active. In the presence of a particular protein kinase, we know protein kinases phosphorylate other proteins, then what happens is you get phosphorylated EIF2, and that is now inactive. So you can turn off EIF2 activity. You can turn it back on. There's phosphatases which will take off phosphates. So the example I'm going to give you of how this regulation takes place physiologically is an example that occurs in developing red blood cells. So in developing red blood cells, basically what they're doing is they are producing a large amount of globin to make hemoglobin. That's really the goal. The blood cells are jam-packed with hemoglobin. So during the development of these red blood cells, they have to synthesize a lot of globin protein. Now, in addition to the globin protein, there's 4 globin protein molecules that come together to make a complex, but in addition to that, you require a heme group. A heme group is kind of it's a cofactor. It's not an amino acid base. It's a more complex organic structure. Very similar, if you remember back to when we talked about photosynthesis, when we looked at chlorophyll, it's that kind of ring structure that has the magnesium in there. In this case, it has iron in there. So it's a larger organic molecule that needs to be there in order to make mature hemoglobin. So what happens is, that has to be synthesized. So what happens is that heme, actually, the level of heme controls whether or not globin protein will be made. So there's no globin translation if there's little or no heme in the cell. So the cell is basically, quote unquote, rationalizing, why should I bother making the globin protein if I can't make the whole hemoglobin because the heme is not present? So heme controls the globin translation. And if there's sufficient heme, then the globin translation will occur. Now, all this activity, the regulation takes place through a protein that's called a heme-controlled inhibitor. A heme-controlled inhibitor. That heme-controlled inhibitor is an EIF2 protein kinase. Like I told you, there's a protein kinase phosphorylates the EIF2. Well, the one we're talking about is actually called EIF2 protein kinase. So it specifically phosphorylates EIF2. And the heme-controlled inhibitor controls the activity of that kinase. So let's look up at the top right, kind of diagrammed it out. So the heme-controlled inhibitor is an active protein kinase. And so it uses ATP, it phosphorylates the EIF2, and so translation is turned off. That happens if there's no heme available. No heme available, we don't want to translate, so phosphorylate EIF2 turns off translation, initiation. If there is sufficient heme, well, it turns out that molecule, that protein, the heme-controlled inhibitor, has a binding site for heme. So heme is an allosteric regulator of the HCI or heme-controlled inhibitor. So if there's sufficient heme in the cell, some of it will bind to the heme-controlled inhibitor, and it will allosterically turn it off. Once it turns it off, then the EIF2 is no longer phosphorylated, and then you can get translation of the globin. So here you have regulation of initiation through modification of EIF2. Now I have written below, just to point out, that this is actually an antiviral mechanism, that cells use this to control viral infections. So I just have it sketched out very simply. So upon viral infection, the cell responds. Sometimes they refer to it as an innate immune response for viral infections. The cell gets infected by the virus, it turns on interferon. Interferon is a protein that activates a kinase which is called PKR. PKR is a protein kinase which has one target, and that target is EIF2. So it'll phosphorylate EIF2 and turn off translation. What's happening here is that when a virus infects a cell, it basically commandeers all the cell machinery to make more of itself. So it commandeers the polymerases, it commandeers the nucleotides, it also commandeers the transfer RNAs and the ribosomes to make protein so it can make more copies of the virus and kill the cell. So here the cell is saying, no, we're going to sacrifice ourselves, we're going to turn off all protein synthesis, and the cell will die, as will the virus. So that's an antiviral mechanism. I want to just say something about this here. This is when you turn off EIF2, you're turning off all protein synthesis. That's what's happening with the interferon. I mean, that's a pretty dramatic activity. You turn off all protein synthesis, the cell basically just stops. The example I gave you with the red blood cells, and the reason why you can afford to turn off EIF2 in those cells, is not I should say, it's not a very commonly occurring way to regulate because it's kind of a brute force thing. But it can happen in the developing red blood cells. It can happen there because that's all that those developing red blood cells are producing. All they're producing is globin. So they're not producing other stuff. They're not dividing. They're not doing much of anything else except producing globin to make hemoglobin. So in that case, you can afford to turn off total protein synthesis because virtually the entire system is synthesizing globin. Okay, so we looked at initiation. We looked a little bit at regulation of initiation through EIF2. And we have looked at elongation. And the last thing that occurs is termination. And we'll end with that. So what happens, actually there's a couple more things I do want to say. What happens during termination is that there is a terminator codon or termination codon on the messenger RNA, and the one they're showing here is UAG. They're showing it by red. And so what they're showing at the top is a peptidyl transfer RNA, which can be basically, well, it will be the entire protein. It could be 1,000 amino acids. And then the ribosome reaches the stop codon in the A site, and then what comes in, there's no transfer RNA. What comes in is called a release factor. The release factor is actually it's a cluster of proteins that fits into the A site, interacts with the the UAG, which is the termination site, and then it basically triggers the termination reaction. What happens in the termination reaction is the whole thing just comes apart. So the protein is released from the transfer RNA. The two ribosomal proteins come apart, and the ribosome is released, and the messenger RNA is released. So it just blows the whole thing up. Now, I do want to say another two things. One is that the cell tries to be as efficient as possible. So with the messenger RNA, often, the majority of the time, it's not one ribosome translating the message. What happens is that once one ribosome starts translating, depends how much protein the cell needs, once the ribosome starts translating that message, then the cap structure and some of the leader sequence is exposed, another ribosome can initiate translation and then continue. And so you can end up getting 4, 6, 8, 10, sometimes even 12 ribosomes translating a message at one time. So it increases the efficiency, you get a lot more protein. So those are referred to as polyribosomes. So here's a drawing of polyribosomes, and just showing multiple ribosomes translating a single message. And you can see the 5' end of the messenger RNA would be the cap, and there's the start codon, so that one just jumped on. And then all the way at the other end by the poly A tail, it's hitting the stop codon, and they're representing that that's the end because the peptide that's coming out is much longer. That's the green. And then as one comes off, it moves down the line, another one can come on. It just depends on how much protein the cell needs. So those are referred to as polyribosomes, multiple ribosomes translating a single message. Okay, so we're going to stop here, and I want to, next time there's a little bit more I want to talk about, because I want to talk about the medical implications of understanding the mechanisms of protein synthesis. Turns out that many antibiotics that we use specifically inhibit specifically inhibit prokaryotic or bacterial translation while leaving eukaryotic translation intact. So you can kill bacteria, leaving our cells okay. We'll talk about that next time. All right.